Hello and welcome back to everyone. My name is Shirsha, and in this particular lecture we are going to talk about immunohistochemistry and immunocytochemistry. So these two techniques are commonly utilized to visualize tissues and cells respectively. First, we will talk about immunohistochemistry in depth, and I will talk to you about the various steps in sample preparation, what are the applications of immunohistochemistry, and what are the merits of this particular technique. After that, we are very briefly going to also talk about immunocytochemistry, how to prepare the sample, and what exactly do we mean by immunocytochemistry. So without further ado, let us dive right into it. Immunohistochemistry. So what is immunohistochemistry? What do we exactly mean by it? Immunohistochemistry or IHC involves the visualization of tissue or cellular components within a tissue by immunostaining. So what do we mean by immunostaining? That is by utilizing antibodies for the purpose of staining. Now, as you can easily guess that since we are going to be using antibodies for staining, so antibodies by nature are highly specific for their specific epitopes. So we would actually be staining specific proteins or specific structures within the tissues or cells in a tissue section for immunostaining. So the main or major application of immunohistochemistry is with respect to studying the upregulation or downregulation of expression of any specific particular protein. So let us assume you have a healthy condition and a disease condition, and you suspect that your protein, let us say protein A, gets upregulated in the disease condition. And this might either be a causative agent for the disease or a manifestation of the disease condition. So one way of studying this is by performing immunohistochemistry. So you assume or you suspect that the expression of your protein gets altered in a specific tissue, let us say in the brain tissue. So then you would prepare brain tissue sections from a healthy individual and brain tissue sections from a diseased individual, and you will stain both of these tissue sections for this specific protein, protein A. You will be using antibody which is specific to protein A, which will ensure the specificity of your staining. And when you compare the two, when you visualize the two, you can then make out if your hypothesis is correct or not. That is, whether there is indeed an upregulation of expression of protein A in the disease condition or not. Now, tissue preparation or fixation is essential for the preservation of cellular morphology as well as tissue architecture. Now, let me be more specific. You expect that within the brain, a specific region of the brain or a specific subset of cells present within the brain exhibit this altered expression of your protein of interest. So in order to be able to pinpoint that, you should be able to preserve the tissue architecture. Otherwise, if the tissue architecture is disrupted during the various steps of sample preparation, then you can't really say for sure where you're observing this altered expression pattern of your protein of interest. Not only is it imperative to maintain the tissue architecture, but you should preserve the cellular morphology as well, because ultimately you will be using antibodies which will recognize specific epitopes on your protein of interest, protein A. So if these epitopes get degraded during the subsequent steps of sample preparation, or if they are somehow altered or masked, then that will defeat the entire purpose of your experiment. So in order to help with the preservation of cellular morphology and tissue architecture, the samples need to be fixed, and it is very, very crucial. Inappropriate or prolonged fixation, if done, may significantly diminish the antibody binding capability. Now, many antigens can be successfully demonstrated in formalin-fixed paraffin-embedded tissue sections. And this is the most common method of preparing tissue sections, where you first embed your specific tissue in paraffin and then you cut it or slice it. Because if you think about it, a tissue is not really very rigid. So when you try to cut it or section it, you don't really want to end up squeezing it out. So let us, for the sake of example, imagine a tomato. So when you're slicing a tomato and if you apply high pressure, then there is every chance that you're really going to end up squishing the tomato and the seeds and the juices start flowing out from inside. So what happens? The tissue architecture is getting disrupted when you're sectioning it. Similar is the case when sectioning any tissue, like say for instance, as we were discussing the brain. So if you want to slice or section the brain, you don't really want to squish the brain during the process of sectioning. So to help avoid that, typically tissues are embedded in paraffin because paraffin is nothing but a type of wax. So it will provide a solid surface which you can easily hold on to, and then you can easily make the sections of the cuts. Now, we will discuss all of these steps of tissue fixation and how to embed your tissue in paraffin and how to section it in detail. I'm just giving you a brief idea of why we do what we do. However, it is important to keep it in mind that some antigens will not survive even moderate amounts of aldehyde fixation. Again, aldehyde is an agent which is commonly used for tissue fixation. But we will discuss about this later in details. So under such conditions, the tissues should be rapidly fresh frozen in liquid nitrogen and then cut with a cryomicrotome. So a cryomicrotome is a special kind of device that we will again talk about. So under the situations where your antigens are very volatile in terms of not being able to survive even mild fixative agents like aldehyde, you should directly go for snap freezing your tissue. Now once frozen solid, the tissue is already hard and everything is frozen in place, and now you can make sections of it. However, the disadvantage of frozen sections include difficulty in cutting compared to paraffin sections and the need for frozen storage, because you can't really let it melt or thaw out, because if it does, it would defeat your entire purpose. It's not like that you can let it thaw and then you can freeze it again. That will again disrupt your tissue architecture and is therefore undesirable. So this is a beautiful picture of a tissue section prepared from breast cancer cells, the ductal carcinoma in situ. So it's an IHC for estrogen receptor which shows positive nuclear staining. So the estrogen receptor is actually stained over here, and as you can see, it is beautifully stained in this specific section. So there are a number of factors that influence the result of your IHC staining. It depends on the tissue handling and how well you're handling your tissue, how careful you are with it or not careful. It depends on tissue fixation. That is, what method of fixation you're using, what chemicals you're using for fixation, because each and every chemical has its own merits and demerits. So tissue fixation also affects the quality of your IHC results. It depends on how you store the sections once you've prepared the sections, because come on, let us be practical. Once you prepare the sections, it is not like you're going to go for imaging on the same day. So how you store your sections plays an important role. It depends on epitope retrieval. This is something that we will again discuss in detail, that what exactly do we mean by epitope retrieval. But let me just briefly explain it to you. So because you're using antibodies for the purpose of staining, antibodies will recognize specific epitopes. So you have to ensure that your epitopes maintain their integrity during the processes of tissue section preparation, and also that they are not somehow masked during the processes of preparation. So these two criteria fall under the umbrella of epitope retrieval, and this also plays an important role in determining the results of your IHC. It obviously depends on the choice of antibody, because that is your stain, that is the stain you're using. It depends on the detection system, because again each and every detection system has its advantages and disadvantages. So whether your antibodies are coupled to enzymes, that is you're going to go for enzymatic detection, or whether your antibodies are coupled to fluorescent molecules, will affect your results. It depends on phosphorylation detection, because now phosphorylation is one of those things which will not survive the typical procedures of tissue fixation. So if you want to study phosphorylation of any protein, then you would have to go for snap freezing of the tissues and then subsequent sectioning using a cryomicrotome. It depends on whether you're doing manual IHC or automated IHC, because this will also fall under the purview of handling, although not really tissue handling, but rather sample handling during the steps of preparation. The results also depend on whether you're interpreting them manually or by using some software. And finally, it depends on the visualization technique used. That is, whether you're going for bright field microscopy or dark field microscopy. So bright field microscopy is when the background of your imaging is bright. So if you are using antibodies that are enzyme conjugated, that is you're going to go for colorimetric based enzymatic detection, then you would go for bright field imaging. And dark field is when your background is dark. So if your antibody is conjugated to a fluorescent molecule and you're going for fluorescence based detection, under that situation you'll be going for dark field microscopy. So all of these factors will influence your IHC and the results of your experiments. So let us now talk about how to prepare samples for IHC. So a general immunohistochemistry protocol consists of four main steps. Well, I'm sorry, that is a typographical error. It doesn't comprise of four, but rather it comprises of six main steps. First, fixation, to keep everything in its place where it was to begin with. Second, paraffin embedding, which will help with sectioning of the tissue, which brings us to our third step, tissue sectioning. Fourth, antigen retrieval, to increase the availability of proteins for detection during staining. Fifth, blocking, to minimize pesky background signals. So the purpose of blocking is to ensure that only your specific protein of interest is bound by the antibody. And finally, last but not the least, antibody labeling and visualization to get the pretty pictures. So if we were to summarize the various steps involved in the sample preparation of IHC, we would begin with the tissue sample. We would then process the tissue, which involves fixation and paraffin embedding. This embedding will give us the tissue block, which we then section using a very sharp blade. This leads to tissue sectioning. Now, once we have the tissue section and we have retrieved the antigens, we are going to block the background and stain the section. And finally, we will visualize our results. So to begin with, we will first talk about the first step of sample preparation for IHC, that is tissue fixation. Fixation preserves morphology, prevents tissue degradation, ensures antigenicity, and helps to protect cells from damage during the tissue preparation process. Because you have to keep in mind that when you have isolated your tissue from the organism, the moment you isolate it, the tissue will start to undergo gradual degradation. It's the same thing because the tissue is now dead. It is no longer a part of the living organism which was keeping the tissue alive. Also, not only is the tissue dead, but several steps are involved in preparing the sample, in processing the sample, in making it susceptible to staining and visualization using specific antibodies. So you don't really want your sample or your tissue to get degraded during these steps either. It also helps preserve morphology, and like we have already discussed, preservation of morphology is essential so that you can pinpoint out exactly where the changes are taking place in expression pattern of specific proteins, because ultimately the entire point of staining a tissue section is not only to visualize where exactly the protein is expressed, but also to be able to compare how this expression pattern might alter under different circumstances. So it is important to preserve the morphology and also to ensure antigenicity. That is, the antigens are available for binding with the antibody paratopes. And it is not that the antigen itself gets degraded during the subsequent steps of processing. Now, the reproducibility and the quality of the IHC results are highly dependent on several factors, which includes fixation time, that is how long you are going to subject your samples to the fixation agent, the temperature of the process, the pH, and the buffer composition of the buffers that are used for fixation. And all of these must be carefully optimized and standardized for different starting materials. You can't really say that, okay, I have this one generic method that I'm going to use for all tissue samples irrespective of the source of the tissue. No, that will not work. This is a very sensitive method and it requires empirical optimization for different tissue samples to yield the best results. So within tissue fixation, we will first talk about selection of the fixing solution. So we will discuss about some of the commonly used fixing solutions, but again, each of these need to be tested to see whether a specific type of solution is appropriate for the specific antigen you want to detect, because there is no standard fixing solution for different kinds of antigen immobilization. So not only will this choice depend on the source of the tissue, that is the type of the tissue being fixed, but it will also depend upon the antigen that you're trying to stain and then visualize. So one of the most commonly used solutions for fixing is acetone and alcohol. Acetone and alcohol are both primary fixing solutions, which play a role in precipitating sugars and fats, as well as maintaining the immunologic competence. What do we mean by immunologic competence? That is the ability of your tissue section to react with specific antibodies. That is its immunogenicity or its antigenicity. Note that alcohol is ineffective in maintaining low molecular weight proteins, polypeptides, and cytoplasmic proteins. So if you are trying to visualize either of these three, then you should steer clear of using alcohol as a fixing solution. However, alcohol can be mixed with glacial acetic acid, ethyl ether, chloroform, and formaldehyde to help overcome this specific downside. Acetone, on the other hand, is often used for frozen tissue and cytological smears. Now, when would you prepare cytological smears? Say, for instance, you want to study certain non-adherent cell types like RBCs or red blood cells. Under such situations, you would prepare a cytological smear. So, cytological meaning cellular and smear, because well, you will just take the blood and make a smear of it on the glass slide and then go for visualization. So why is acetone used for these two? Since it has a strong penetrability and dehydration property. So for frozen tissue, these two criteria become very important, because you have to keep in mind that your tissue is actually in a frozen condition. So its cell membrane or cell wall, depending on if it is from a plant source, but mostly IHC is done with tissues isolated from animals. So the cell membrane, well, that is now frozen solid. So you need a fixing solution which can penetrate through the frozen cell membrane and should have good dehydration property. So acetone is suited for using with frozen tissue. The next solution that we are going to talk about is aldehyde. Aldehydes are difunctional cross-linking agents. So difunctional meaning that is it has two different functional groups which can help in cross-linking proteins. So why is it a cross-linking agent? Because one of these functional groups will interact with one of the proteins and the other functional group can interact with either another protein or well with a different amino acid residue within the same protein. But because it has two functional groups, so it is difunctional. And it is widely used because of its strong penetrability, low contractibility. That is, the tissue doesn't really contract when subjected to aldehyde treatment, and low background. Now, it is very important that in order to get a crisp signal, the signal to noise ratio should be high. And in order for that to be so, the noise, that is the background, should be low. So aldehyde provides a cross-linking between tissues and also helps maintain the antigen. While formalin, which is a saturated 37% formaldehyde solution, is the most widely used, 4% paraformaldehyde is better than formaldehyde as it is the pure form of formaldehyde and methanol free. And methanol would otherwise interfere with the fixing process. The final kind of solution or fixing solution that we are going to talk about are non-aldehydes. So this includes carbodiamide, dimethyl acetamide, dimethyl subirimidate, and parabenzoquinone, all of which are widely used for the fixation of peptide hormones in tissues and are better mixed with glutaric dialdehyde or paraformaldehyde. Again, all of these details are irrelevant unless and until you do a literature survey on how the specific tissue that you're trying to stain has been stained in the past. So it is very important to do a complete background research, and if it is a new tissue, then you have to be ready to try an array of different fixing conditions and sample preparation conditions that will help you to identify the best condition to get the optimal results. So next we are going to talk about the different fixation methods. Now the different fixation methods are more generic as compared to the different fixing solutions that one can use. The number of fixing or the number of different fixing solutions available is quite vast and they have to be tested. However, fixation methods are quite limited and you can use only one of these available fixation methods. So let us talk about that. So first, we're going to talk about fixation by perfusion. Now, what exactly do we mean by it? As fixation can cause cross-linking. So if you remember, we talked in the previous slide about how aldehydes are bifunctional cross-linkers. So this cross-linking can mask the epitope. So the best way to preserve tissue morphology as well as target protein antigenicity is to replace the animal's entire systemic blood volume by a fixative with vascular perfusion. That is, you drain the animal of its blood and instead of the blood running through the animal's blood vessels, you have the fixating agent or the fixative solution running through the animal's blood circulation. This is followed by perfusion with a sucrose solution. So this will mask or this will fix all of the tissues that will come in contact with the circulation of the animal. The next technique which is commonly used for fixation and it is more commonly used than fixation by perfusion is fixation by immersion. So you can actually make out what it would entail from the name. So for majority of applications, it is not really feasible to perform fixation through perfusion. You can't really afford to replace an organism's entire blood circulation with your fixing solution. It is just not done. You can only do it for very small laboratory animals. What about samples that you get from diseased patients, or what about autopsy samples? So an alternative method to that is to immerse the tissues in fixative solution, and you can do this to tissues isolated or obtained from any source. Biopsy and surgical specimens commonly employ this fixation method. The fixation duration is generally determined by the antigen's stability and also by the type of fixing solution that is being used. So both these factors, that is the antigen's stability and the type of fixing solution used, will play an important role in determining the duration of fixation. And again, this is something that also has to be optimized. The third and final method for tissue fixation is snap freezing. So we have already briefly discussed about the kind of samples that do indeed need to be snap frozen. So for cases in which standard fixation techniques cannot be used or fixation will be carried out later, tissues can be snap frozen and kept at -80 degrees centigrade until use. So let us say you have obtained your tissue from a biopsy patient and you are not really going to process it right now. So you snap freeze it and you store it for later use. Because snap freezing will help in ensuring that the tissue integrity is maintained and protein degradation is avoided. Also, for those tissues where, for instance, you want to observe any changes in the phosphorylation pattern. So there also, because phosphorylation is a tricky alteration to detect using immunostaining, and normal methods of fixation will result in either destruction of the signal or masking of the signal. So under such situations also, you would go for snap freezing of the sample. So frozen tissues are prepared by immersing the tissue in liquid nitrogen or isopentane, or by simply burying the sample in dry ice. Now, if you immerse them in liquid nitrogen or isopentane, that will result in snap freezing. That is because the temperature will drop drastically and immediately, it will immediately freeze everything, ensuring that the overall distribution of proteins and other cellular components remains unimpaired and unaltered. Snap freezing is frequently used when detecting post-translational modifications like phosphorylation. So this is something that we've already discussed. And because now it is important, why? Because changes in phosphorylation pattern of proteins affects how a specific protein is functional. We are well aware that in different signaling pathways, majority of signaling pathways are regulated by either upregulation or downregulation of protein phosphorylation, because this simple post-translational modification is sufficient to alter a protein's functionality. So therefore, detecting post-translational modifications sometimes becomes very important. And under such situations, you have to go for snap freezing of the samples. The frozen tissue is then cut using a cryostat or a cryomicrotome. These tissue sections may then be fixed, typically with an alcohol such as methanol or ethanol. So in case of the other kinds of tissue samples, first you will fix the tissue and then you will go for tissue sectioning. But for snap freezing, first you freeze the tissue and then you section it and fix it after you have sectioned it. So the order over here is getting reversed. Now, since alcohols do not mask epitopes, so their use will circumvent the requirement for antigen retrieval, because the entire point of antigen retrieval is to unmask those epitopes which might get masked during the process of fixation. So if you are using alcohol, this particular step of antigen retrieval can be avoided. So this is what a cryomicrotome or a cryostat looks like. So as I have already previously mentioned once, because your tissue is frozen and you don't really want to thaw out the tissue during the different steps of processing, including sectioning. So in this cryostat, the temperature inside is maintained at such that your tissue samples will remain frozen. So it is cooled inside. So there is basically a glass window over here, which you can't really make out in the image. So the glass window slides up. You put your tissue inside for sectioning. There's a super sharp blade over here that will make very thin slices of your tissue. And when you put your sample in, you then close this chamber. So the duration for which you keep this open is not really long, because you want the inside temperature to be maintained. And sectioning is done under cold temperature conditions to ensure that the tissue does not thaw out. So next, we're going to talk about some of the precautions that you need to take when fixing tissue samples. So these are some points that you should keep in mind when fixing tissue samples. So exercise caution when fixating tissues. Number one, do not overfix the tissues. No, overfixation is not at all good for your sample, and it will only mar the results that you are trying to obtain. Store the tissues appropriately after fixation, because it is not necessary that you will process the tissues the same day that you fix them, or you will go for visualization the same day that you fix the tissue. So it is necessary to ensure that the tissues once fixed are appropriately stored. And if you can recall, this is one of the factors that we covered in under factors influencing IHC. Use adequate amounts of fixation solution. Do not be skimpy with it, and wash sufficiently after fixation. This washing becomes important to ensure avoidance of overfixation, because if you are not careful with the washing and if you're not rigorous enough with it, then some amount of the fixative agent might remain associated with your tissue, and this will lead to overfixation. Also, reduce the tissue sample size to less than a specific dimension. This is like very specific, and it's applicable only to those who are performing IHC. In case you're not really doing IHC and you are here only because you want to learn about the mechanistic basis, the different steps involved just for the sake of knowledge, then you can avoid this. But what you can keep on mind is that the tissue size or the sample size should not be too large, because if it's too large or if it's too thick, then that will impair with the ability of the fixing agents to penetrate well into the depths of the sample of the tissue and to fix it. So if possible, reduce the tissue sample size. If however it is such that your tissue is such that you can't really reduce its thickness without compromising the integrity of it, then don't go for it. You can find a way around it using different fixing solutions and durations and conditions and so on. So this brings us to the second step of sample preparation for immunohistochemistry, and that is paraffin embedding. So embedding the tissue in paraffin is required for the next subsequent step, which is sectioning. So how exactly is it done? I'm going to briefly take you through the steps involved in paraffin embedding of tissues. So the process of paraffin embedding involves fixation, dehydration, transparentizing, immersion, and embedding. So I will take you through each of these terms individually and what they mean. So fixation is something that we have already covered in depth in terms of commonly used fixing solutions and the common methods of fixation. So once the tissue has been fixed, it needs to be dehydrated. Why? Because tissues or cells contain a lot of water. As human beings and animals are made up of 70% water. And paraffin is a wax. It is hydrophobic or water hating. So your paraffin will not really go into the tissues if there is water inside it, because the water will repel the paraffin. So it is important to dehydrate the tissues or to remove the water from the tissues. So this dehydration process generally removes water and hardens the tissue. After dehydration, the tissue of interest requires a transparentizing step, because the dehydrating agent used in the previous step is immiscible with the paraffin in the following steps. So not only is the water present inside immiscible with the paraffin, but the dehydrating agent that is used is also immiscible with the paraffin. And this removal of dehydrating agent is called transparentizing, which will make the tissues amenable to entry of paraffin into the tissue depths. So the addition of a transparent reagent helps the paraffin absorption into the tissue. Commonly used transparentizing agents include xylene, benzene, and chloroform. After transparentizing, the tissue can now be immersed in molten paraffin wax, allowing the absorption of the wax substituting transparent agent. So the wax will now enter into the tissue and it will displace out the transparentizing agent that is present within the tissue. And now your tissue is filled up with wax. So based upon the melting point of wax, immersion should be performed at 54 to 64 degrees centigrade. So within this range, typically your paraffin wax is molten and your tissue can be immersed into the paraffin. And once the dehydrated and transparentized tissue is immersed in the molten wax, the wax will enter into every crevice and every nook and cranny of the tissue. Next comes embedding. So during the process of embedding, the paraffin infiltrated samples from the previous step are individually enclosed in mesh cassettes with a specific orientation in the middle of the block for subsequent sectioning. So once you have paraffin infiltration into your tissue, you take this paraffin infiltrated tissue and you enclose it in specific cassettes and you keep it in a specific orientation. So your tissue, when it's in the molten wax, when it's in the immersion step, at this stage also it has not yet solidified, like the wax has not yet solidified. So you can take the tissue out and you can place it in whatever orientation you want within the mesh cassette. Now this orientation becomes important during sectioning. So depending on whether you want horizontal sections or vertical sections or sections at a particular angle, so you will position the tissue accordingly within these cassettes. Now the equipment that you'll be using for sectioning, that is a microtome, it has a blade that cuts at a specific angle, and you should be able to, and it also has a specific holder where you'll be placing your tissue, paraffin embedded tissue. So typically your paraffin embedded tissue will be in a block. So in what angle the tissue is present within the block of paraffin will determine at what angle the tissue will get sliced, or at what angle tissue sections you will obtain. So this becomes important. Following this, the cassette is filled up with molten paraffin and it is left to cool down. After cooling is completed, the tissue will now be ready for sectioning and suitable for storage. So if you go through the steps of paraffin embedding, so first you fix the tissue, you rinse it thoroughly and vigorously to remove any trace amount of fixation agent. Then you dehydrate it. After dehydration, you clear it or transparentize it, and then you immerse it in the wax. So all of these steps are carried out in jars or special jars, in which you just, you have the different solutions in the different jars, like the fixing solution, the rinsing solution, the dehydration, clearing solution, and finally your molten wax. and you dip the tissue in one, keep it for the required amount of time, take it out, dip in the other, keep it for the requisite time, take it out, and so on and so forth. Now, once you have immersed your sample in wax, you have to place it in this cassette, and the cassette is filled up with the molten wax. So like for example, here we're talking about the brain. So the brain is placed, well, the right way up. Maybe you could rotate it 90 degrees, okay, and you can place it, or you can place it at a specific angle in between the horizontal and the vertical position, depending on at what angle you want to section the tissue. And you let it cool down. Addition of cold water helps with the cooling procedure. It quickens it. And now finally, when you take it out of the cassette, you get a block of wax, and within the wax embedded, you have your tissue of interest. And next, you will go for sectioning of the tissue. There is a specific blade in the microtome which will cut out extremely thin sections. So like I said, the blade is fixed inside the microtome at a specific angle, and it also has a holder where you'll be placing this block. So how you place the tissue inside the cassette will determine at what angle it gets solidified within, and at what angle you're getting the tissue sections. So like here you're getting vertical tissue sections. If you had flipped it over here at 90 degrees, then you will be getting horizontal tissue sections. So I hope that is clear and I have conveyed what I want to say. So moving on, next we are going to talk very briefly about tissue sectioning. So after the tissue is embedded in paraffin, it is cut into thin slices or sections. Paraffin embedded tissues are sectioned using a microtome. So this is what a typical microtome looks like. So you see, you have the blade here, you have the holder, and here you will collect out the tissue sections as it's coming out. And this is an example of a tissue being sectioned. So you see, the holder is holding this block of tissue embedded in paraffin. So you can't really change the angle at which this block is being held in place, right? And there is a sharp blade over here, it's not really clearly visible, which is passing through this, making super thin slices. You see, extremely thin slices of your tissue which is embedded in paraffin. So I would like to reiterate that how you place this tissue within the block will determine the angle of the sections and what kind of sections you're going to obtain. So you see, to begin with, you had this tissue embedded in thick wax, and now you have super thin slices of the same. And you can go for staining and visualization of this. So next we are going to talk about antigen retrieval. So antigen retrieval is an important and necessary step to ensure that your epitopes are available for binding by the antibody once you add the antibody and when you're staining the samples. So formaldehyde fixation usually generates methylene bridges which cross-link proteins and therefore mask the epitope of interest. So if you can recall, or in case you cannot, if you go back to the initial slides where we were talking about the different fixing agents, we mentioned that formaldehyde is a difunctional cross-linker. So when it cross-links these proteins, it will generate methylene bridges in between the or within the amino acid residues of the proteins, which will cross-link the protein, and this might end up masking your epitope of interest. It is essential to therefore recover the antibody epitopes in order to allow the antibodies to bind. So this can be done by two methods. The first of them is by heating the samples. So this is referred to as heat-induced epitope retrieval, or here. That is how I pronounce it. It might be different from the way of pronouncing it. So in that case, just ignore my pronunciation. So the HIER method can be implemented by microwave, high pressure, or using a water bath. It will break these methylene bridges formed by formaldehyde and expose the epitopes to allow the antibodies to bind by continuous heating. So heating will disrupt these methylene bridges. So whatever cross-linking had taken place would now be undone, and whatever epitope was getting masked would now be exposed and available for antibody binding. Comparing to the other method, Pierre, which we are going to discuss about, here has a gentler experimental condition in which users have more control over the experimental parameters. Because ultimately what you're going to do is you're simply going to heat your sample. So at what temperature you're heating, how long you're heating, you can exercise a lot of control over that. However, heating is not just done randomly, but rather in a specific buffer. It's mostly a citrate buffer solution, which contains stress and EDTA and some other components. We're not going to get into that, which will help with the disruption of the methylene bridges. However, the pH and the buffers that are going to be used for HIER required to be optimized. Okay. So you have your slide with tissue. You cover it with a plain empty slide. You basically overlap these two slides. You use a paper clip to hold them in position. So your tissue is not really directly exposed. It is covered by the other slide. And you immerse it in boiling buffer and you heat it. This heating will help break open or break apart the methylene bridges. So the second method for antigen retrieval includes enzymatic digestion, and this is called proteolytic induced epitope retrieval, or PIER or PIER. Again, this is the pronunciation that I'm used to. In case you call it something else, ignore my pronunciation. So epitopes can be exposed by incubation with proteases. So what are proteases? They're enzymes that cleave proteins, which will enzymatically digest the methylene bridges. However, you should take care that your entire aim of treating your samples with proteases is to expose your epitope, which was masked. So you wouldn't really want to subject it to excessive proteolytic cleavage such that the protease ends up cleaving or degrading your epitope itself. So that has to be avoided under all circumstances. The choice for digestion enzyme used depends on the antigenic components. Pepsin and bromelain are used for retrieving antigens in intercellular areas. Epitopes can be exposed by incubation with proteases, which will enzymatically digest the methylene bridges. Other enzymes can be used for intracellular antigen exposure. So whether you're looking for intercellular antigen or intracellular antigen will determine what enzymes you will be using for the PIER procedure. It is suitable for retrieving more difficult epitopes, while the pH for incubation is usually known. So you know at what pH you really need to incubate your samples. However, it is definitely a harsher method and can damage your tissue morphology, because ultimately you are using a protease and your epitopes are also proteins in nature. So therefore, this method involves the risk of damaging your epitope of interest as well as the tissue morphology, and it is a much more harsh method compared to the milder and gentler HIER. And personally, I think HIER should be your first method of choice. However, if it's some difficult antigen that is getting masked and it's difficult to expose it, so difficult antigen in terms that it's difficult to retrieve it using heat, then you have to go for proteolytic cleavage. So this image will help clear it up. So you have, see, the blue section is the epitope where your red antibody molecule is supposed to bind, but it can't really reach it because all of these methylene bridges, see the CH2 bridges, are masking it. So you add your enzyme, it cleaves somewhere here, let us assume, and this part is now removed and your epitope is exposed to the antibody for access. However, keep in mind, the enzymes that you're using, they are not really that specific enzymes that they will cleave only at a specific sequence of amino acid residues. They will cleave after specific amino acid residues, right? So there is every possibility that it might end up cleaving somewhere here, and your epitope is also getting displaced, like this protein peptide fragment, let us say, which is getting removed here, only that contains your epitope. So this is something you have to keep in mind, and you have to be aware of the protein's amino acid composition, and you should make an informed decision about what enzyme you're going to use for proteolytic digestion. So this brings us to our next step, which is blocking. Now, blocking is something that we have already dealt with before in previous lectures on this channel when talking about ELISA and Western blotting. So both of these techniques, ELISA as well as Western blotting, utilize antibodies which are specific for specific proteins whose presence we want to detect. So we know that antibodies are highly specific for their antigen, and they will only specifically bind to it. But there is always the possibility that it might not specifically interact with other empty vacant sites on your sample. So for instance, in case of Western blotting, you have your protein of interest on these PVDF membranes. What about the rest of the membrane where your protein is not there? Because the membrane binds to proteins non-specifically, so when you will add the antibody, it will bind to the empty membrane. So under such situations, blocking becomes imperative to ensure that your antibody has only specific regions, or rather your antibody has access to only these specific regions where your epitope might be to bind to it, and it can't really bind to the background. I hope I'm making sense. So the activity of HRP, that is horseradish peroxidase or alkaline phosphatase, used for signal visualization, requires blocking or inhibition of endogenous enzymes as well to avoid non-specific binding and hence artifactual signal. Now this is something new to us, the inhibition of endogenous enzymes. So because we'll be dealing with tissues, so tissues have what? They contain cells, and cells can contain these enzymes like horseradish peroxidase and alkaline phosphatase naturally. So these, when you add the substrate, the endogenous enzyme would also interact with the substrate. It will react with it and it will give some background signal. So it is very, very important to inhibit the endogenous enzymes to avoid this non-specific background, which will only bring down the signal to noise ratio and reduce the quality of your images. So residual sites on the tissue section may bind to secondary antibody, given we are going for indirect method of detection, and produce follow-up false positive results. Therefore, serum from the same species as the secondary antibody is commonly used for blocking those sites. So let us, for an instance, assume that we are doing indirect labeling and we are using a primary and a secondary antibody. So for the purpose of blocking, you utilize serum from the same species from whom the secondary antibody is prepared. So if you are using secondary antibodies isolated from goat, then you will use serum from goat for the purpose of blocking to avoid the cross reactivity between the blocking agent and the secondary antibody. So this will help in ensuring, why? Because since the secondary antibody is from the same species, so this will help in ensuring that the serum is non-reactive or inert, just like any blocking agent should be. So the blocking should be carried out at room temperature for 10 to at most 30 minutes to avoid excessive blocking. Okay. Again, excess of nothing is good, neither fixation nor blocking. So if you go through the sections of sample prep, I'm not sorry, if you go through the steps of sample preparation for IHC one by one, so this is something we've already dealt with. That is, you prepare the tissue sections and undertake any antigen retrieval or endogenous enzyme inhibition. And then you move on to blocking with 1 to 2% of serum isolated from the same species from whom the secondary antibody is being used. you add primary antibody, and so you prepare the samples, you block it, you then incubate it with primary antibody for approximately 30 to 60 minutes. So this is the labeling step. You then wash it for 3 times, 5 minutes each. Washing is done to ensure that any antibody that is sticking non-specifically gets removed or washed off. Then you add a secondary antibody and again incubate it for another 30 to 60 minutes. You wash it again thrice to remove any secondary antibody that is non-specifically associated with it. Now you go for addition of the substrate, considering that your secondary antibody is coupled with an enzyme. So you're adding the enzyme's substrate. Again, you wash it to remove excess substrate. And finally, you develop the signal and you visualize it. So now we are very briefly going to talk about the different staining techniques. This is something that we have already covered in detail when talking about Western blotting. So I'm just going to, for those of you who are new to the channel and for those of you who have not gone through those previous videos, I'll be just very briefly touching upon these topics. So visualization of signal can be done using either fluorescence-based detection method or enzymatic detection method. So antibody staining in IHC can be either direct or indirect. So for fluorescence-based detection method, your antibody will be coupled to a fluorophore, and for enzymatic detection method, your antibody will be coupled to an enzyme. So coming to direct method of staining, so what do we exactly have in it? In direct methods, the primary antibody is directly conjugated with a fluorochrome or with an enzyme. Cells or tissue sections are incubated with the primary antibody. Unbound primary antibody is then removed by washing. The sections are mounted and viewed under a microscope. So you will go for a bright field microscopy if you are using enzymatic detection method, or you will go for dark field microscopy if you are using a fluorescence based detection method. If you are going for enzymatic detection method, after the unbound primary antibody is washed away, you have to add the substrate and let it develop, and only then will you go for visualization. And if it's conjugated to a fluorochrome, in that case, after washing away the excess antibody, you can directly go for visualization. So this is what it is like. You have these antigens, you have the antibody against it, it is coupled to the enzyme or the fluorochrome, you add the substrate, you get the signal. If it's coupled to the fluorochrome, then you irradiate it with the light of specific wavelength and you get fluorescence. And in the last lecture uploaded on this channel, we talked about immunofluorescence. So we talked about how exactly fluorescence works, you know, the Jablonski diagram and everything. What is the chemistry behind it? So next we have indirect method of staining. So in indirect methods, the primary antibody is unlabeled and is detected with an additional fluorochrome or enzyme labeled secondary antibody, right? So cells or tissue sections are first incubated with the primary antibody. Unbound primary antibody is then removed by washing and excess antibody is removed by washing. Sections are then incubated with the secondary antibody, which is coupled to the fluorochrome or the enzyme. Typically, the secondary antibody is a fluorochrome or enzyme conjugated anti-isotype antibody. So this is something again we have already discussed in previous lectures. So anti-isotype antibody, what do we mean by it? So let us say your primary antibody was raised in rabbits. So the Fc portion of the antibody is, that is the constant portion of the antibody, will be conserved within a specific isotype. So let us say you have rabbit IgG, right? So your secondary antibody will be against the Fc portion of rabbit IgG. So it's an anti-isotype antibody which will bind to the species specific portion of the primary antibody. So the FAB parts of antibody that is specific for the epitope, and it is not really conserved within a species. It Whereas the Fc portion for a given isotype of antibody, it is conserved in a species. So the secondary antibody is then removed by washing, and the sections are mounted and viewed under a microscope. Now, if you are using enzymatic detection method, so some of the commonly used enzyme labels include HRP or horseradish peroxidase. So a reaction of horseradish peroxidase with hydrogen peroxide and DAP, that is diaminobenzidine, yields a dark brown alcohol insoluble product. So the image that we saw in the initial slides, I think the second or the third slide, that is of enzymatic detection. That is a result of DAP staining. Okay. So in this case, if you're going for enzymatic detection, then you will view the slides under a light microscope or a bright field microscope. So this is the indirect IHC. You have the antigen, you have the primary antibody against it. So this is the FAB portion of the antibody, these two, and this is the FC portion of the antibody. So this is conserved within a species for a specific isotype. So the IgG, for instance, is an isotype. And the secondary antibody is specific for this Fc portion, and this is now conjugated to either your enzyme or your fluorochrome. So we have already talked about the advantages and disadvantages of direct and indirect labeling techniques when talking about different staining techniques in Western blotting. So if you are new to the channel and if you have not gone through those lectures, I would suggest that you go back to the lecture on Western blotting and learn about the differences between the two and the advantages of one over the other, because that is not something that we are going to cover in this particular lecture. So indirect staining methods may also involve the use of avidin biotin labeling and ABC labeling. So in avidin biotin labeling, what do we have? So you will have a biotinylated secondary antibody, an enzyme which is labeled to avidin. So it will just go and associate with the biotin on the secondary antibody. Or you can go for ABC labeling. So what exactly do we have in ABC labeling? So in avidin biotin complex labeling, you have the primary antibody, which is specific to your antigen of interest, and the primary antibody is unlabeled. It does not have any labels on it. Next, you're going to add the biotinylated secondary antibody. So the secondary antibody will recognize the Fc portion of the primary antibody and bind to it. Next, you're going to form an ABC or avidin biotinylated enzyme complex. So your enzyme will actually be bound to streptavidin. Now, streptavidin can bind to multiple avidin molecules. So this will ensure that one primary antibody can bind to multiple enzyme molecules. So this will actually amplify your signal, and when you finally add the substrate, you will have a more intense signal at the point. I hope it's clear what I meant. If you have any doubts or if it's not clear, then do leave a query in the comment section and I will try to clarify it better. So next, we are going to very briefly talk about multiple labeling experiments. So in order to do multiple labeling experiments, you cannot really go for enzymatic detection. Why? Because, well, the enzyme that you're going to use will be a peroxidase or a phosphatase. So the difference in color of the product produced after the enzyme interacts with the substrate will not really be much, or perhaps you don't really have so much variation in it, which will allow it to differentiate between two different antigens that have been labeled. So if you're going for labeling of multiple epitopes or multiple proteins in one shot, then your antibodies have to be coupled to fluorophores and you have to go for fluorescence based detection. Because why? Only in immunofluorescence is it possible to visualize multiple proteins simultaneously, provided that your fluorophores emit lights at different wavelengths, that is, of different colors. So the degree of spectral overlap between the two fluorophores that will occur when using several fluorochromes at the same time should be taken into consideration. The lower the spectral overlap between the two, the better your signal would be. So what do we exactly mean by spectral overlap? You wouldn't really want that you're using two fluorophores, say A and B, and the excitation wavelength of fluorophore B overlaps with the emission wavelength of fluorophore A. Under such situations, what would happen is that whatever signal fluorophore A is emitting, some of it you can detect and a lot of it would be taken up by fluorophore B to excite fluorophore B. So this spectral overlap should be minimal so that whatever is being emitted by one of the fluorophores is not being sequestered by the other fluorophore, and you can visualize emission from both fluorophores efficiently. Okay? So the excitation filter is composed of a set of filters which transmit light over a narrow band of wavelength. So this is something we have again discussed under immunofluorescence. In single labeling, typically long pass filters are used, which are designed to maximize detection of emission over a wider range of wavelengths. Multipass filters, which allow more than one antigen to be visualized simultaneously, can be used to visualize distribution of antigens. Why? Because again, in immunofluorescence, we have discussed about excitation and emission filters. So I'm not really going to get into that over here. But when you're using multipass filters, they will allow light of multiple wavelengths to pass through to the detection system. So this will allow the visualization of more than one antigens simultaneously. However, multipass filters do result in some loss of brightness and may cause a signal to become undetectable if two coincident signals are viewed where one signal is much dimmer compared to the other. So if one of the fluorophores or one of the signals is much more intense compared to the other one, it can end up masking the other one. Anti-fade reagents like citifluor can also be applied to the sample to reduce photobleaching. Again, what is photobleaching, how it is detrimental is something we have covered previously, so I'm not really going to discuss it over here. So this is an image of HEK cells stained with M cherry and M YFP. So M cherry fluoresces red and M YFP fluoresces green. So the entire purpose of multiple labeling experiments is to study the co-localization of two proteins. So here you see in this channel you can see the red like the red colored protein where it is localized, not the red colored protein but the protein which is stained with M cherry, and here you can visualize the protein stained with MYFP, and when you overlap the two, you see in the nucleus only you will see the green signal, but in the other regions you can see an overlap of both the red and the green signals, which suggests that these two proteins are co-localizing in these specific vesicles that are being studied over here. And I can proudly say that this image has been taken from one of the papers published from our laboratory itself. So yeah, there's that. So finally, we are very briefly going to touch upon immunocytochemistry. And in fact, the previous image or the image in the previous slide is an example of not IHC but of ICC, because you're visualizing individual cells over there and not whole tissue sections. So immunocytochemistry or ICC involves visualization of cellular components by immunostaining. In contrast to IHC, ICC utilizes cells that are grown within a cell culture. So again, this is where your knowledge on mammalian cell culture systems will become useful. Cells are usually fixed using formalin. After fixation, the cells or tissues are permeabilized. Why do we have to go for permeabilization? To allow the antibodies to permeate within the cells so that we can now stain intracellular proteins. So this is usually done using a non-ionic detergent like Triton X100, which increases membrane ruffling and pore size. Or if you want to go a non-detergent pathway, then you can utilize acetone, which opens up membrane pores and may even partially rupture the membrane. Permeabilization is necessary to allow antibodies to enter into the cell. So it is an important and crucial step. So this is an example of ICC where you can see that individual proteins within the cells are labeled, such as the TH in the axons of sympathetic autonomic neurons. And in blue, you have the nuclei which have been stained. So the blue stain is DAPI. It is a common counter stain that is utilized. So if we briefly go through the steps, you begin with the cells. So either you can culture the cells in transparent glassware or on cover slips taken on glass slides, or you can prepare smears if you are dealing with non-adherent cells. You fix it. The logical reasoning behind fixation remaining the same as in IHC. You can either go for chemical cross-linking, for example, using formaldehyde. Again, we have talked about how it works. Or you can use precipitants like methanol or acetone. So if you're going for chemical cross-linking, then permeabilization becomes imperative. However, if you're using precipitants, then these also permeabilize the cells. So there is no need to use an additional permeabilizing reagent or detergent. And once you've permeabilized, you block it using serum. Next, you go for incubation with the antibody. So if you're going for direct immunofluorescence detection, then your primary antibody that you're going to use is already labeled. If you're going for indirect detection, then first you will incubate with an unlabeled primary antibody, and then you will add a secondary antibody which is labeled with the fluorophore. And then you counter stain. Now, why is counter staining important? So counter staining will typically either stain the DNA or the some cytoskeletal elements. So this will give you an idea as to the orientation of your cells. Because here in immunocytochemistry, you will not be going for enzymatic detection. You will be going for fluorescence-based detection. And in dark field microscopy, it is good to have a counter stain because that will give you an idea of the outline of your cell. So for instance, if you're counter staining for the nucleus, then you know how many cells are present, what is the distribution of the cells, where the cells are. So the counter stain is typically against the DNA, which is present in each and every cell, or against some cytoskeletal elements, which is again conserved among different cells. So you can use a common counter stain. Okay. And then if you're like for instance growing the cells on cover slips, then you have to go for mounting. Or if you're growing the cells in transparent culture ware, then you can avoid this step of mounting and you can directly use this culture ware for visualization. You can just place it in the holder on the microscope. And finally, you go for microscopic analysis. So you see over here some beautiful pictures of ICC, the nuclei which is stained with DAPI, and here you can see that this red fluorophore, this is phycoerythrin, again something that we have talked about in the previous lecture, which is staining the actin cytoskeleton of the cells. And here is another beautiful picture of ICC, just to give you an idea of how the cells actually look. So you see it's dark field microscopy. The background is black. And these are all examples of multiple labeling experiments. So spectral overlap is minimized, and spectral overlap is something that has to be kept in mind when you're going for multiple labeling experiments. Now some key points to keep in mind. One of the main difficulties with IHC or ICC staining is overcoming either specific or non-specific background. cells or tissues are treated with solutions of an inert protein for blocking, right? For example, bovine serum albumin or fat-free milk or serum from the animal from whom the secondary antibody is isolated for indirect labeling in case of IHC. Additionally, positive controls and negative controls are also essential for determining specificity of your antibodies. And this is something that we always keep in Western blotting experiments and ELISA experiments as well. IHC and ICC can be performed using fluorescent dyes, that is, using immunofluorescence, enzymatic methods, using enzymes such as peroxidase, or even by autoradiography. Although this is less and less common these days, owing to the negative impact of radiation. The choice of labeling usually lies between a fluorescent label or a colored product of an enzymatic reaction. Although other labels like colloidal gold ferritin can also be employed for immunocytochemistry. So these are some links that you can go to if you are interested in learning more about IHC and ICC. So the first two links are for IHC and the last one is for ICC. So it talks about the different steps and the different reagents that you use for all of the steps, and also an in-depth protocol based on like the time durations of each step and so on and so forth. So if you are interested for knowing more, then you can always go to these links. So this brings us to the end of a very elaborate discussion. So this is again an example of IHC, where you can see DAP staining has produced this brown product which is staining these structures over here specifically. So to summarize, we talked about IHC in depth. We talked about what it is, what are the different factors influencing IHC, what are the different steps involved in sample preparation, what needs to be kept on mind, and what are the different conditions that have to be taken into account. We then talked about ICC very briefly, what it exactly means. We also talked about the different detection techniques. There is direct detection, indirect detection, and ABC labeling mediated detection. We also very briefly touched upon multiple labeling experiments and what are the key points that need to be kept in mind, and what are the advantages of doing it, what are the problems associated with doing multiple labeling, and what are the key applications of multiple labeling experiments. So that brings us to the end of this particular lecture. I hope you found this useful. In case you have any questions, any doubts, any queries, please feel free to leave it in the comment section and I will definitely get back to you. If you like this video, then do like it and share it. And if you are interested in more such content on different techniques commonly used in laboratories, then do subscribe to this channel for more such content. So that is it, and I will see you again next time. Bye.